pre-signed in. That's good. I like that. Ooh. Okay, so now to go back here. All right, and we're live, you guys. It's not, it'll be on Facebook in a second. There is a delay if you didn't know about the delay. So, okay. Hey everybody, welcome to Science Colloquium for February 10th. I'm glad you guys are here today with us. Our talk today on COVID-19 will begin at 3:15 p.m. Just before we get started, I do want to tell you about our next upcoming event, which will be February 24th. Again, that'll be 3:15 on that Wednesday. And we have Gianna Amador, the co-founder and managing director of Carbon 118. The title of her talk is Science Policy Innovation Action. And is it possible to create a world where climate change and economic prosperity are drivers of innovation? The discussion will cover innovation and implementation by farmers, foresters, and it's funny on my screen, I can't see, and business to reduce as well as sequester carbon from our atmosphere. Gianna Amador is going to share her experiences as an advocate for carbon sequestration. And so that's in 2 weeks for Science Colloquium. February 19th, coming up next week, Modesto Area Partners in Science, which is also brought to you from a lot of folks at MJC. We're going to have Justin Fish at 7:30 p.m. That's Friday, February 19th. And his title of his talk is A Land Dedicated to Science, the Antarctic Treaty and its Environmental Protocol. That's going to be a Zoom virtual event that will also stream live to YouTube. They have a little different protocol how they do things at MAPS. So if you want to get the Zoom link a day before the event, you can sign up on the MJC webpage that I have at the bottom. So it's www.mjc.edu/instruction/SME/MAPS. So sign up for that, and we hope to see you there. Today we are going to be looking at COVID-19 and doing a little update. I will be talking on one part. My name is Elizabeth McInnes, and I teach biology and botany here at MJC. And we also have...
I am Deborah Martin, an anatomy and physiology instructor from MJC.
And I'm Erin Lucas, the microbiology instructor here at MJC. And I also, in addition to teaching Micro 101 for the pre-nursing folks, I also teach Plagues of Mankind, which is kind of apropos in our whole situation that we have going on right now. So I'm going to share my screen and let's get started. Maybe if it'll share. Here we go. All the buttons. Okay, so my part of this discussion today focuses on vaccines and kind of where we are with vaccines. But before we start our vaccine chat, which is going to be kind of brief because there are 3 of us and we each have a topic, it always behooves us to take a peek at just our basic biology and our basic virology knowledge from before. So if you remember your biology class, viruses are acellular. These are not living things, right? So it is important to remember they don't have an agenda. They aren't out to get us insofar as there's any meaning behind it. They're not vindictive. They're essentially sacks of genetic material of some kind, whether it be DNA like cellular life or whether it be RNA. And that genetic material is surrounded by a protein coat. And sometimes there is also something called an envelope, like an example is SARS-CoV-2. And these things kind of bump into host cells. And then when they bump into host cells, if it's a cell that they can infect, it'll gain entry into that host cell, and then it'll be replicated within that one cell, oftentimes using a lot of host cell machinery to do that. And so you get a lot of viral multiplication, and eventually those viruses leave the cell and go on to infect other cells that are neighboring. And so you start to get symptoms of disease when enough cells are involved and enough downstream things start to happen. So if we kind of look at the most basic schematic of this of all time, if we have just this regular cell, it's any kind of a cell. If we're talking about SARS-CoV-2, it's probably an upper airway cell, or somewhere, if you take a really deep breath, it might be a lung cell. And the virus is going to come into contact with that cell. Then there's going to be some receptor binding. So there's a protein on the surface of the virus, and that's going to connect with the protein on the surface of the cell. And that's going to be actually Liz McGinnis's talk. So we're going to leave that. It gains entry into this cell where the envelope, if it's enveloped, comes off, the protein coat comes off, and you're left with just free nucleic acid. So again, if we're thinking about SARS-CoV-2, this is in the form of RNA, and it's roughly about 30,000 bases long, which is either big or little, depending on your point of reference. That nucleic acid is then going to be replicated inside the cell. Then we get new viruses made. That happens, and eventually they leave the cell to go on and infect other cells. So kind of with that in mind, right, let's look at SARS-CoV-2. And a little point of nomenclature here. In the news, it's called coronavirus. And while that's true, it is a coronavirus. There's actually hundreds of coronaviruses out there. That's a large group of viruses. Some of them infect people, some of them infect animals. We've known about coronaviruses for a while now. We have 4 coronaviruses that cause the common cold in people. And so they've kind of been on our radar. But importantly, we've actually had twice in our history coronaviruses that cause major disease in people. Those being SARS-CoV-1 or SARS Classic, depending on where you get your nomenclature, which caused the SARS outbreak in 2002, and then MERS-CoV, which is the coronavirus that caused the MERS outbreak in 2012. So we've known about these things for a while, but now we are on to SARS-CoV-2, which is the name of the virus, the specific coronavirus that happens to cause the disease we have then named COVID-19, right? Okay, I hope everybody's coming with me on that. So why do I bring all that stuff up? It has to do with vaccination and, what are our goals? What's the purpose of vaccination in general? And the fact that we've had some history with these coronaviruses that cause severe disease will come up in a minute. But when we think about vaccination, right, what is the goal or the purpose of vaccination? Is that we want vaccinated people to have protection, and we want that protection to be durable or to last, right? So we want immunological memory. Now, there's not enough time to do a full immunology lecture here, so let's just start parsing this out as far as what is our ultimate goal. That word protection, we want vaccinated people to be protected, is nuanced. Protected from what? We have to be specific. We should be specific. Ideally, in the absolute perfect world on the perfect planet and everything is amazing, we want protection from infection, right? We call that sterilizing immunity. If you have protection from infection, that means that you could take a full-blown cough laden with SARS-CoV-2 droplets right in your face, and your cells don't even become infected. They do not multiply the virus, right? And if they don't multiply the virus, you have no transmission, you have no disease whatsoever, and that would be absolutely ideal and perfect. But we don't want to throw out the good and the very, very good in search of perfect. So what do we ultimately want, right, with vaccination? We want protection from disease. And then you have to kind of parse that out too. Well, are we talking any kind of symptomatic COVID-19? Or do we want to keep people out of the hospital, keep them out of the ICU, off of ventilators? Do we want to prevent death? And the answer is yes, we would like to have all of those things. And we have, a lot of those things. So let's talk about the actual vaccine candidates that are either available now or coming through the pipeline. I'm actually going to talk about 4. You guys all probably know about the Pfizer and the Moderna vaccines. Those are the mRNA platforms. Johnson & Johnson has formally requested EUA authorization. The A is redundant. And the FDA will rule on that by, I think, late February 26th. And then I actually think Novavax is kind of on the heels of that and the next in the pipeline. So we're going to mention Novavax as well. With the mRNA vaccines, you probably have heard this before, right? This is a vaccine platform. Both the Pfizer and Moderna platforms are very similar. It includes the genetic code, so not any part of SARS-CoV-2 itself, but the genetic code for either the full-length spike protein. We're not going to talk about spike protein. I'll let Liz do that. Or in the case of Pfizer, it's actually just a piece of the spike protein called the RBD, receptor binding domain. That's the part that actually comes into contact with the receptors on our cells. And in those 2 platforms, that bit of genetic material is encoded, is encapsulated in a little bit of lipid. So that goes into the syringe. When you get your jab, the idea is that the muscle cells near the injection site are going to be able to take up that platform, that messenger RNA, and then the machinery inside your host cells, the ribosomes, are going to translate that into either the RBD of spike protein or the full spike protein, and then those cells are going to display it to the immune system. And then you will get, hopefully, a robust B cell response, so an antibody response, and a robust cellular response or a T cell response. And both of those are really amazing. And we get that protection. With the Johnson & Johnson platform, it's a little bit different, but kind of also a little bit the same. What Johnson & Johnson has done is they have taken the genetic code for spike protein. And instead of just using just that, they have stuck that genetic code in an adenovirus vector. What that means is there's a group of viruses called adenoviruses. In this particular case, it's adenovirus 26 or AD26. And when they've put that genetic code into that adenovirus, AD26, they've also removed the part of the virus that is involved with replication. So that means that this is a replication deficient or unable to replicate viral vector. So they take that, the genetic code for spike in the AD26 backbone, put that into the syringe, you get your inoculation. Same type of a thing is supposed to happen here. The local muscle cells should uptake that, and they will be able, with those host cell machinery, to translate that spike protein, show it to the immune system. You get a B cell or an antibody response, you get a T cell or cellular immune response, and you're good to go. Novavax is also very exciting, and they're very clever. This is a protein subunit vaccine platform. So what Novavax has done is they've taken insect cells in culture. So these aren't like bugs, but rather just insect cells. And they have convinced these insect cells to make the spike protein. So they use these cells as essentially factories to make a bunch of spike protein. And so then they take that, just the protein, use that in the syringe, you get your jab, and those localized immune cells then will have the protein, see the protein, become reactive to it. You should get a robust B cell response. You should get a robust T cell response. So when we look at those different platforms, and we say, okay, let's compare them. This is where sometimes the news gets a little bit, they give you some numbers, they don't give you all the numbers, they don't tell you what the numbers mean. That's unfortunate. When you look at the different vaccine platforms, 3 of these are a 2-dose system. So you get one inoculation, then you have to go back, get another one. J&J has a 1-dose platform. You get your jab and then you're good. Well, then you wait a while you mount an immune response, and then you're good to go. When you look at efficacy, right, how good they are, this is where I have asterisks under Pfizer and Moderna because they're wildly amazing, right? But they did their phase 3 trial and they reported from pre-variant times. The variants have always existed, but it wasn't dominant until now. It's kind of becoming dominant. And their efficacy for preventing symptomatic COVID is amazing. 94 and 95% effective at preventing symptomatic COVID. And when you look at J&J and Novavax, they've actually done their phase 3 trials kind of in this variant time. And so they have data that goes with both kind of, the SARS-CoV-2 information and then also the variants as well. And J&J, they came out and said, okay, 4 weeks after your vaccination, in areas where the variant was not seen very much, we have 72% efficacy at preventing mild or moderate disease. And you might be like, oh, that's not very good. 72% is really, really good under normal considerations. The only reason we don't think it's amazing is because Pfizer and Moderna came back with bigger numbers. But remember, these are just different. J&J also said, because they have some of their phase 3 places of interest were in areas where the variants were dominant. And so they have variant data. And it does look that with the variants, 4 weeks after vaccination, you get a little bit decrease of efficacy. Okay, fine. Novavax had about the same kind of a thing. If you were infected with a non-variant SARS-CoV-2, great efficacy. If you're infected with a variant, less so. And so people might be like, I don't know then, maybe they're not that great. But let's really think about what do we want out of our vaccines? What's the goal? The goal is to prevent people from dying. The goal is to keep them out of the ICU, keep them off ventilators. And when you look at all of these vaccines, they are all amazing at preventing severe disease and preventing death. And I put on there J&J after day 49. It actually takes a little bit of time for you to really mount a strong immune response. So it takes a bit of time. So it's not just 2 weeks out after your vaccine there. But all of these vaccines, all 4 platforms are really, really, really good. And just to assuage, I get a lot of questions of, oh, this vaccine is rushed. I don't want to take it because it was rushed. It's not safe. Let's just talk about kind of how this happens, right? Normally, if you're thinking about making a vaccine to something, you have a lot of preclinical study. Those are the petri dishes in the lab, maybe some animal experiments. If that looks good, you go into phase 1. If that looks good, you go into phase 2. If that looks good, you do a really big phase 3 trial. You get you're liking your data. You're going to send that package off to the FDA. The FDA is going to take forever to look at it because it's a lot of data and they read all of it. Then they're going to maybe approve you. And if they approve you, then you go on to manufacturing and then distribution. And that's a good business model, right? If it's your money and you don't want to spend money making things that you don't know if you're going to get approval for. But in a pandemic situation, if you have warp speed type money, you can kind of do all of these things and kind of like the same. You can have your preclinical study. Remember when I said we've known about severe disease causing coronaviruses since 2002? Well, let me tell you, there was a MERS vaccine already in clinical trial. And so it was pretty easy, it's all relative, to take that information and convert it over to SARS-CoV-2 information once we got the protein for spike worked out. You can kind of look at phase 1 and phase 2 together. Importantly, you can start manufacturing. Here's where the warp speed money really helped. They started building the manufacturing before they got approval from the FDA. That way, when we got approval from the FDA, really, really, really quickly, they were able to start vaccinating people. So within 18 hours, I think, of FDA approval, needles were going into arms. And it's important to know that when you look at this, there were no steps omitted. All of the safety steps were there. All of the clinical trials were there. They just were able to stack them because they were using somebody else's money. So the answer to all of the questions are you take whatever vaccine is offered to you first. Don't wait. Don't miss your shot. Get vaccinated. And so with that, I'm going to turn it over to Elizabeth McGinnis to tell you all about Spike. Liz, you're muted.
Thank you. I'm always muted. It's a thing. Thank you. I'm excited for a vaccine. So, all right. So, kind of going forward here, you guys, I want to talk a little bit about evolution and SARS-CoV-2 and where we're at now. And so I'm going to start off just a little bit, pick up where Aaron left off, and I want to talk about ACE2. So it's the angiotensin converting enzyme that we have in our cells, and its job is to modulate angiotensin 2, which then lowers blood pressure. And this particular receptor, the ACE2 receptor, which for us it's an enzyme, it's in the tissues of the respiratory system, it's in the heart, it's in blood vessels, kidneys, livers, your digestive system, it's everywhere. And so this is the molecule that the SARS-CoV-2 spike protein binds to, and that's how it gets entry. Ultimately, it's kind of the gatekeeper. It gets entry into the cell for infection. So it's the receptor in this situation. So little bit, I'll tell you, when we originally were planning this, I wanted to talk mostly about animals, bats and pangolins and cats and minks, because we were hearing a lot about how these were acting as reservoirs for virus. But then there's a lot in the news about variants, and that's an evolution story too. So I'm going to concentrate on that. Hopefully that maybe we can come back to in the future. The interesting thing, though, is the ACE2 binding region of spike is highly conserved in mammals. There's little change in shape between mammals. And so there's a high possibility of binding of SARS-CoV-2 between species. And we know it jumped from a species and came to humans. And then it's like, okay, well, can we pass it between humans? And this one definitely we can, we know that. Early on in the pandemic, you might have heard stories about the cats at the Bronx Zoo. Cats were infected with SARS-CoV-2 from somebody working in the zoo. The cats never got the severe form of disease. No cats died, but they were moderately sick. So it wasn't instant concern with cats maybe that are in zoos all over, that especially protected species. We didn't want them all to get SARS-CoV-2 and die. And then you might have also heard about mink in Denmark. There were millions of minks that they culled because there was a pretty horrific infection that swept through, And most of the animals had severe disease and were dying. So they kind of wiped out all of that production. All of those mink farms were kind of taken away. There was one more thing about this. Early on, people were kind of upset that we were using tests to, a lot of us couldn't get a test, right? And in New York, people couldn't get a test, and we were testing cats. Veterinarians actually had prepared those tests specifically, and so no human tests were wasted in that. But it's important we know what's going on in these animal populations because they are a reservoir of virus and a place where virus is evolving. So let's take a closer look at spike. The SARS-CoV-2 virus is covered with this corona of spike proteins. So when you look at it, you can see all of these proteins sticking out. It's a transmembrane protein that is anchored in the membrane of the virus, and it binds to the ACE2 receptor of target cells. And the spike protein itself ends up being a dominant target of our immune system, and it's what we're targeting with vaccines. A little more on spike composition. We can see that this particular protein has different subunits. It has the top subunits, the S1s, and the lower ones, S2. So the top part, the S2s are up here, and they have the receptor binding domain in them. So we're always kind of really interested in what's going on with those proteins specifically in the whole spike protein. And we basically have these, there's 3 proteins that make up a trimer. So then there ends up being 3 receptor sites on a spike. And how do we know all this? Well, we know it from sequencing data. So there's labs all over sequencing, whole genome sequencing the SARS-CoV-2 virus. And they are also using computer modeling and then cryo-electron microscopy. And so this is a cryo-electron micrograph that gives us a nice view of those spike proteins. So want to talk a little bit about variant evolution and some worries that we have because of this. And I'm going to show you some lineages later on, but the virus has been evolving the whole time from the very beginning. And we knew it was evolving. It's just that once there are variants that become worrisome that, hey, are these particular variants where they have changes on the spike protein, is there going to be an increase of efficiency in transmission? So is the binding ability, the affinity between spike and the ACE2 receptor, is it going to be greater? Which for SARS-CoV-2, that mutation would be a good mutation for the virus itself. And then will vaccines still work? If we get too much change, is that going to change that receptor binding domain and then the vaccine doesn't work as well? And then will the treatments that we're currently using, are those going to work as well? And then one big thing that came up is, well, if it's changing this much, how are the tests going to fare with these variants? And so far, it seems like the tests work great with the variants. There hasn't been significant change enough to have an impact on testing, which is great news. So mutations can be good, neutral, and bad. And it's kind of funny when we look at ourselves from our point of view and the virus's point of view, not that it really has a point of view. The mutations that are good for SARS-CoV-2, they're not so good for us. The neutral ones, fine. And the ones that are bad for SARS-CoV-2 are probably pretty good for us. So what are the current variants of interest that we're hearing about in the news right now, and how are they spreading across the planet? We have the one that was first identified in the UK, which is the B117. We have one from South Africa, and we have another one from Brazil, the P1, that was first identified in Japan from travelers from Brazil. There's also another one of interest from California, but we don't have as much information about it yet, so I'm not going to talk about it here today. I'm going to concentrate just to give you an idea of what kind of changes are happening in the spike protein. I'm going to look at B117. Right now from data, it looks like the transmission rate of this is 35 to 45% higher than the original reference Wuhan strain of the SARS-CoV-2. So that's a little bit worrisome. And that's a matter of how many particles is it going to take to give you an infectious dose of the virus. It doesn't mean like somehow this is catching the wind better and getting from person A to person B. It's that more viral particles are going to stick to ACE2 receptors ultimately. So good reason for continuing masking, double masking, and lowering the particles that could ever get into your respiratory system. So this is already spread across 30 states. First, that's between November 20 and January 21st. So this is looking now like this will be the dominant variant in the United States by March. There's a great paper that was out just this Sunday. I'll put it in the show notes, but this one has a very quick 7% kind of increase in its dominance every single week. And so we're seeing it become more and more dominant throughout the United States. So this picture, I want to give credit to the New York Times because I got this from them. It's just gorgeous, but it shows the messenger RNA of corona. And I want to talk just right here about the section, which is 1273, ultimately going to code for 1273 amino acids for the spike protein. And there are 2 amino acid deletions that are in this section. And then there's also 6 substitutions. So we're going to take a look just real quick at the H69 to V70 and the N501Y mutation. Before I do that, I do want to mention here, these are all the amino acids that you would see for the spike protein. And each letter on this page represents an amino acid. So it's not showing a 3-letter codon for an amino acid. It's shortened it up and it has 1 letter for an amino acid. And it's kind of taken away the codon idea in this sequencing. So it has methionine at the beginning going all the way through. But you can see the 2 deletions marked and the single amino acid mutations that you see take place there. And that's where you have a switch in amino acid. So the picture that I have here is looking at the spike protein, kind of looking down from above at the top of the spike protein. And these little spots here are the binding domains. And so if we look at this particular mutation, it is occurring at those binding domains, and it's happening at, sorry about that, at protein, at amino acid 501. And at that spot, an asparagine is being switched out for a tyrosine. So when we look at this name of this mutation, this is showing asparagine It's at 501, so that's the amino acid in that sequence, and Y is tyrosine. So that's the first one. And that's one of the ones we worry that it increases the affinity for the ACE2 receptor. The second one is the H69V70 deletion. This one's worrisome too. It also increases the ability to infect more cells. So there's an increased affinity between the spike protein and the ACE2 receptor. And when looking at this, research is showing that it's evolved from people with chronic infections. So people who have, you hear about them being sick for several weeks. They keep testing positive. They're a reservoir for the virus. This particular deletion has popped up in their samples quite often. So we've also seen it as a recurrent deletion region repeatedly in various coronavirus lineages, both human and animal. It occurred in the Denmark mink outbreak. So it's one to watch. It's one scientists are aware of, and they pay pretty close attention to that one. All right. And then the last thing I just kind of want to mention today is variant monitoring and distribution. There are two groups that are keeping track of data, and the two groups are NextStrain and GISAID, and they're offering platforms where whole genome sequencing of samples, the data from that can be uploaded and then shared. And then it gets organized into various lineages. And at the very center of this, if I had more time, I would link to it because it's kind of fun. I have the website here and I will have it listed in the notes. But at the very base in here, you can click on this and it shows the Wuhan reference strain. And as you move out through here in this radial tree, you can see different mutations that have taken place along the way in these different lineages. So it's a great view of the SARS-CoV-2 from the Wuhan strain to the wide variety of variants that we have. All of those represent a variant. And then as you get further out to the outside, these are the most recent variants, and they've left a little room for February and March out here at the edges. So I will stop there. I have a couple of slides that I'd love to share at the end if we have time left over. Thank you. And I believe Deborah's up next.
Here I am. Oops. Oops. So, hold on a second. So, again, welcome to Science Colloquium. My name is Deborah Martin. I teach anatomy and physiology, and I will be talking about therapy because I'm a physician. I was born and raised in Cuba, went to med school in Cuba, and now I teach anatomy and physiology for MJC. And in my talk today, I will talk a little bit about an update in terms of the therapy of COVID-19. First of all, I want to tell you that the spectrum of therapies, it's growing rapidly and evolving really fast. So in my talk, I'm going to be talking about what helps, what doesn't, and hopefully give you some advice as a physician on what you should do not to get sick, right? Or if you get sick, what you can do. So another point that I want to make is that most people who become ill with COVID-19 will be able to recover at home. And what do? You're going to be doing some of the same things that you do when you get the flu. You are going to rest, right? You are going to keep yourself well hydrated. and you are going to take some medications to keep fever, pains, aches away, like Tylenol, acetaminophen, right? There was a lot of controversy about these two medications. Now the WHO is saying it's fine, you can take either one, just to let you know in case you are a little bit confused about it. So then, beyond that, then the CDC, the National Institute of Health, and the FDA has also advised and authorized some treatment or treatments that might help people that get sick, people that have moderate or severe symptoms, people that require then hospitalization. And then all of these organizations have also authorized treatments for people that have moderate symptoms that might not require hospitalization, but they are patients that are at risk because they have a secondary health condition, because they are obese, because they are children. So, and that's what these organizations are in charge of advising, recommending, looking at clinical trials of different type of treatments, and then advising doctors, health professionals on what to do. So then the treatments are divided into three types: antiviral treatments, then we have immune-based treatments, and then we have adjunctive treatments. So antiviral treatments, it's medications that are going to be triggering the RNA of these viruses so they don't reproduce. Immune-based treatments would be, for example, I don't know if you have heard about monoclonal antibodies therapy. We have, for example, blood products treatments, so some kind of treatments that we use plasma from people that got sick with COVID-19 and have antibodies, high number of antibodies in their bloodstream. And then we have treatments that are going to be every time a person goes to the hospital with COVID-19, we have basic treatment for them, like, for example, anticoagulants. That would be that type of adjunctive treatment. So then we have, so right now, you guys are going to be able to see in this graph how the CDC, the National Institute of Health, the FDA has divided these diseases into different severities. So it's divided into mild symptoms and then moderate symptoms, and if the patients require hospitalization or not. And based on that, then physicians, health professionals are able to decide the type of treatment that is recommended. And again, all of these treatments are based on science. That's important for you to remember, that they are all based on clinical trials that have been done. And again, all of this is evolving. So then what I'm going to be talking about right now, the first type of immune therapy that I'm going to be talking about is an immune therapy that will help patients, I was going to say students, so patients, right, that are not in a hospital, but they are at risk of getting severe symptoms. They are at risk of being hospitalized. And one of the treatments that it's there is using monoclonal antibodies. So when we look at these are different combinations, and probably you heard about this one, this is the combination of monoclonal antibodies that our ex-president got, and he promoted as a cure for COVID-19, which is not true. So all of these are man-made antibodies that mimic what we create our bodies are able to produce by cells responsible for doing that and to fight off different types of pathogens. So these type of monoclonal antibodies are given intravenously. Some of them require just one dose, and patients will receive them in a clinic or a hospital. So it's only indicated recommended for patients that have mild to moderate COVID-19 symptoms that are adult, also children that are 12 and older, and also highly recommended for people that are 60 years old, 65 years old or older, and have other comorbidities, obesity, hypertension, et cetera, et cetera, et cetera. And what these monoclonal antibodies will be doing is attacking the coronavirus spike protein to make it more difficult for the virus to attach to human cells and attack them. So that's, here you see the coronavirus, and here we see some of these man-made, lab-made antibodies. So now, in terms of the treatment for severe COVID-19 patients, people that are having respiratory difficulties, people that require a ventilator, then we have different types of therapies. One of them is the usage of corticosteroids, like these medication that you see right here, dexamethasone. So this, it's recommended for very ill COVID-19 patients, and it has been used since the pandemic began. And it makes a lot of biological sense because these patients have developed a hyperimmune response, what you heard probably in the media about cytokine storm. And then these are going to be steroids, corticosteroids, that are going to be reducing that response of the immune system that went in hyperdrive and producing damaging organs around the body, like the lungs. And that's why we see pneumonia and other type of symptoms related to organs being damaged. This is a medication that is very available. It's not very expensive. Then we have antiviral medications like the remdesivir, and this is a type of antiviral medication that it's given to Ebola patients, but it has been used to treat COVID-19 because it interferes with one of the enzymes that the virus needs to replicate RNA. So this antiviral medication has speed up the recovery time for patients with COVID-19. And it could be given to adults, children, again, 12 and older, and patients need to be hospitalized. Then we have other combinations of medication, a medication like this that is going to be acting on inhibiting an enzyme, the Janus kinase, that will be helping. This medication inhibits that enzyme, and as a result of that, it prevents cellular immune activation and the inflammation process that happens because of COVID-19. Then we have, as an adjunctive therapy, we have every patient that goes to the hospital with COVID-19 and is presenting these kind of severe symptoms, they receive blood thinners like heparin. So, and another type, next I'm going to be talking about another type of therapy, immune therapy, that where here we it's derived from blood products. So it's used among hospitalized patients early in the disease, within the first 3 days after the symptoms appear, and or patients that have problems with their immunity, producing antibodies. So this type of blood product is derived from, it's extracted from patients that again suffer from COVID-19, and they need to have one of the criteria for that plasma to be transfused to a COVID patient that is sick, is that it has to have a high level of antibodies. So these have been a type of immune therapy that just as early as January 2021, the New England Journal of Medicine published an article saying, yes, that patients that receive this type of therapy were 48% less likely to develop severe symptoms. So it seems to help. And here we see, in the next slide, we see how that convalescent plasma is obtained from patients that got sick, they recover, they become donors, and then we transfuse. So this type of, it's important, one of the criteria is that to match that plasma to a sick patient, it has to be a blood type match and gets injected, and then these immunoglobulins will be then competing with the receptors, so the ACE receptors that Liz was talking about, and then binding to these receptors and preventing then the virus to binding to the ACE receptors. So if that way, just preventing, making the disease to go into a not a severe case. And what doesn't work? You heard a lot last year about hydroxychloroquine. And let me tell you, it doesn't work. Here we have other therapies that don't work. So the National Institute of Health, the FDA have made very clear that hydroxychloroquine will not work. Does nothing to improve the health of COVID-19 patients. So please don't take it. And here we have other types of therapies that have been demonstrated that they don't work. You can read about it in the links that I provided for you through the CDC and the FDA. And then I want to tell you something that, like always, right? People has been seriously harmed. Some people have died during this pandemic after taking products that are not approved by the CDC, the FDA, right, and the National Institute of Health. And the FDA has established a cross-agency task force dedicated to monitor people like this, people like this physician assistant. I don't know if you read about it in the news last week. She's a recently elected lawmaker from the state of Missouri, and she started selling amniotic fluid, saying that it has stem cells. She earned about almost $200,000 by selling these to her patients. She paid only $251 for a liter of amniotic fluid. So please, my message to you, you should not take any medicine to treat or prevent COVID-19 unless it has been prescribed by a healthcare professional that you trust. Is it clear? So, And then my final message to you is what's ahead. Number one, get vaccinated. Please get vaccinated. Yes, 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 yes. Vaccines alone are not enough to fight COVID-19. So it will take years to get that vaccine on someone's arm. We need to work on developing and discovering new treatments. But for now, what you can do is please, infection prevention. Try not to get sick. How can you do it? Follow his example, right? Follow instructions from people that know. Like here we have Fauci, right? Wear a mask, right? Wash your hands. Don't go to crowded places. Stay 6 feet apart from other people. Listen to Fauci. Listen to science. Thank you so much.
Thanks, Deborah. That was awesome.
Yay. All right, so we got a number of questions in the chat. And I think if it's okay with you, gals, if we just take them one at a time.
Yeah.
Starting from the top. So Ivan asked right off the bat, this is a good question. What about long-term side effects of the vaccine since we've only been vaccinating people a very short time? And that's a great question. When you have, so way back, well, I say way back, it's all relative, right? We've only done this for a year. Back when the FDA kind of was negotiating was putting forth their guidelines as to how they were going to accept vaccines for EUA, one of the time points, and actually this was a really critical time point, they said is, we will not evaluate any phase 3 trial data before 2 months after the second shot. And the reason they picked that as a time point is because the vast majority of side effects from the vaccine are going to happen within the first 2 months. And actually, most of them are going to happen within the first week. So the FDA said, just to make sure, let's pick that 2-month time point. And that's the minimum. Now, with that, those clinical trials now started in September, second dose, I think like very early on in October. And so we're talking now we have from October to February, we actually have 4 months worth of data. And so far those things look good as well. So I'm wildly optimistic that we're not going to see tremendous, something really off the walls somewhere. I'm not at all concerned about some of the really crazy things that are out there. And related to that, what about boosters, right? Because as we've heard, Liz was talking about the variants. So variants arise just kind of randomly, and also when there's selective pressure put on, and that will always happen. So right now, let me just tell you, so right now, if what Liz's research, and I'm with Liz on this one, the B117 variant is going to become the dominant variant in this country and probably a lot of other countries as well. Not to worry, these vaccines have efficacy against that variant. So I'm really confident that if that's the one that goes forward, then it's going to be a-okay. The South African, I hate talking about these from their geographical locations. It's actually really inappropriate.
The B1351.
The B1351 variant. First isolated in South Africa, does show a little bit of what we call escape, which means that the vaccines are not quite as efficacious against that. But I'll remind you that still, so far, the data is maybe that could be true with respect to some kind of symptomatic COVID, but they're still wildly effective against preventing death and preventing severe disease. And so now it's just going to be, do we need to update our vaccines as different mutations arise that lead to different types of, escape? And the answer is maybe. It's possible. I will say one benefit of some of these platforms is that it's really pretty easy to quickly change parameters, especially the mRNA vaccines. All you have to do is change the genetic code slightly, do some cursory safety checks to make sure it still is okay, and then get those out. So I'm really encouraged as far as that goes.
Yeah.
Hope that answers your question.
Well, maybe it's too hard. I did have one little slide on that, and kind of fun, like looking at these variants. So far, all the lab tests, they're working. So that's
yeah. And the vaccines so far are working, even if their efficacy for mild to moderate disease is a little bit less. It will suck if I become infected. It's going to suck if I have to stay home for a week because I have flu-like symptoms or something like that.
Right.
That won't be fun. I want to stay out of the ICU. I want to not be ventilated. I want to not die. So like I said before, I don't want to throw out the good, searching for the perfect.
I think that's pretty good.
Let's see. So are we good on that one? Good on that one? Okay. Yeah. Rosalinda asked, does having previous SARS infection confer, well, I'm paraphrasing. Does having previous infection with SARS classic or SARS-CoV-1 confer protective immunity against this one? So the answer in any life science question is always a gray area, but I will say that SARS classic in 2002, when it popped up, killed a lot of people, major outbreak, very scary. That being said, it has not been seen since 2005. And it infected a relatively small number of people, some 8,000 individuals or 8,200 individuals. So if you're one of those 8,200 individuals, the answer is maybe. So they have been able to look back at some frozen samples from patients from before. And when they tested it at that blood at, I think, 5 years, they showed still good immunity. They looked at 10 years, maybe not so much, but you can't really tell if the lower amount of antibodies they have now will be related to protection going forward. That's not something that I can tell you right off the bat there.
Yeah.
And then JC, what about the allergic reaction to what can the allergic reaction against the mRNA vaccines be attributed to? Ooh, that's a good one. First, if you're nervous, let me just say that those anaphylactic responses are only in 0.00001% of the people that have been vaccinated. So it's very, 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 very rare. And with that, allergies are one of those things where an immune system is kind of hyperactive. And so if you have an immune system that is in generally very hyperactive against certain things, that's where we get allergies. So I'm not sure if it's known what component exactly in those vaccines that are triggering that reaction. But as long as you aren't, like, if you don't have an anaphylactic reaction to previous vaccines or something like that, a major serious allergy, you shouldn't have to worry about those anaphylactic issues. I will say that is one of the reasons where they make you sit there for 15 minutes just to monitor you to make sure you're going to be okay. And they have the epinephrine just in case something goes awry.
Well, and it wouldn't be, they're not allergic to messenger RNA.
No. I mean, you can't be, if you're allergic to messenger RNA, you're probably going to die because you got a lot of messenger RNA in all of your cells.
So, there's a nice little broth that's stabilizing that messenger RNA, and there's something in there. And I bet a lot of those lists are kind of locked away behind the doors of
proprietary information.
Yeah, and Moderna. But I bet that they are gonna hit that hard in some way.
100%.
They're gonna research that. So probably at some point we'll get some data and maybe the names of some chemicals that we can kind of go, okay, that's the thing. Maybe.
I will say, for those that, if you know somebody that is highly allergic to whatever vaccines, the rules might be different with the Johnson & Johnson vaccine because that's a different platform and it's just one single dose.
Yeah.
And so far with what they've put forth, their data, there was zero severe allergic reactions like that. So it could be something to consider.
Yeah. Did you want me to?
Yeah, you've got the other JC question.
Yeah, I think he threw another.
Oh, yeah, he did. It was something to the effect of, do we know why some people get severe COVID rather than mild COVID?
Yeah, and you know what's funny? The reason I was kind of excited about this question is there was an email today from 23andMe. If anybody knows anything about 23andMe, they take a genetic sample and they give you a little outlook on what your traits are and what your probabilities are for certain medical conditions. I did my dad's a few years ago. I didn't want to do mine because I didn't want my DNA floating out there, but I did my dad's. So I get all his emails. And it's really interesting. So 23andMe is doing a study and they are basically data trolling all of the people who have donated samples to them and paid to donate samples to them, by the way, which is kind of fun. And they're trying to figure that out. But, right now we don't know. Some people early on were like, get your blood type checked. It's not that. So it's interesting. They're kind of trying to look at the data they have and ask their users hey, did you have COVID-19 and how sick were you? Were you hospitalized? And trying to see if they can find some sort of link in there. I think we'll find something out about that going forward years in the future, but there could be the other thing that it's not a genetic thing at all, and that it's how much of a dose, how much initial virus were you infected with, how many cells had their ACE2 receptors bind to SARS-CoV-2 real quick, and
related to that too, where was did most of the virus hit you in the nose and stay in the epithelial of your nose rather than deep, did you take that deep breath right at the wrong time and the virus actually started a primary infection in your lung cells?
Right.
It's probably going to be a combination of all of those things.
I think it's going to be something like that. And that's just stuff we're still running the experiment on, It'll be fun when we get to find out.
So a related question, I'll just throw it in there for just a very direct. Rosalinda asked later, does blood type factor into severity of disease? And I know there was a couple of papers that said, hey, I'm linking up this blood type, but if you look at the real big meta papers that have come out in immunology, they really don't show any kind of link at all. So sorry if you have the right blood type. Okay, so let's see. I answered one of Alexandra's questions in the chat, but I'm going to give you just a quick 2-second rundown of the different tests and what they're useful for. We wanted originally to include that information, but there's just so much to talk about. This could go on for hours and hours. The nose, so the deep into your brain all the way back nasal swab, right? The nasopharyngeal swab that you may or may not have had taken. That test, as well as some of just the regular nose swabs, test for the presence of the genetic material of the virus. That's called a PCR test. And what that does is they take that sample, they add some reagents, put it into a machine. The machine will say yes or no to, are there genetic markers of SARS-CoV-2 in that sample? If there are, you're positive. If they're not, you're negative. And that has really good sensitivity to, low levels of infection. So even if you're not symptomatic, if you're very early on in infection, if you're very late in infection, that can be a really good test to kind of look at that. But there's some, it takes forever, it's expensive. There are also some nasal swabs that are called antigen tests. So if it behooves you when you go and get tested to ask what test you're being tested for. If you have an antigen test, that is a test that's looking for specific proteins on the virus surface. I'm a wildly huge fan of those tests. However, I'm huge because you can generally do those at home. You don't need any special stuff for it. The issue with those tests is that you need a pretty high viral load for those to have really good numbers as far as picking out positives that are positive. So if you're low, if you're very early in infection or very late in infection and you just get one of those antigen tests, it may not show you the true picture, but it's pretty good at telling you whether or not you're infectious, like your viral load is so high that you might be infectious to, your neighbor. So I'm a fan of those, and I'd like to believe in the future we get lots of those to where we can each take one of those tests once a day if we want to, because it'll be super cheap. But anyways, the antibody test, some of you guys have gotten a finger stick, and that is an antibody test. That is not looking for the virus at all. That is looking for your antibody. So that's looking, essentially, you had an infection before. You mounted a natural immune response to it. And part of that natural immune response are antibodies you made against the virus. So when you get a finger stick, that's looking for your antibodies against the virus. And there's 1,000 pluses and minuses to the antibody test that could be a whole talk in and of itself. So if you have further questions on that, just email me and I'll give you the long answer for that.
I like this question, and if anybody else wants to take it, jump in, but what normal reactions could appear after the vaccine?
I like it.
Wait, I want to tell.
Okay, you go.
No, and I haven't been vaccinated yet, so I do want to hear I want to hear your experience. But I will say my husband was vaccinated in December and he was doing great. Everything was fine. Vaccine was over, and he doesn't get sick very often. And he's never the guy that's cold. So he went about 2 weeks ago, got his second shot, and within a couple hours was on the sofa, and shivering, cold. And he had a fever. And
wait, was that the first or the second shot?
The second dose.
Yeah, the second one, I went hunting with your husband that day and he was fine. It was 24 hours later.
No, he was sick that night.
Yeah.
He was okay the next day. He was feeling kind of fighting fit by the end of the next day. But that first night, I was like, oh, you need to go to bed.
Yes.
But, yeah.
So okay. So with the 2 mRNA vaccines that are on there, for the first shot, the first dose, people are reporting, your arm is sore. I've yet to have a vaccine in my life where my arm didn't get sore. But things like headache, some muscle ache, some chills, things like that are not terribly uncommon. People, they're usually resolved with a little bit of Tylenol. I guess we should ask the physician. Usually resolved with a little bit of Tylenol or ibuprofen, and within a couple of days, if anything, if you do have some reactogenicity, you're good to go. The second shot does seem to have whatever your reaction was the first time. The second shot is like that, but a bit worse. So if your arm was sore, it's going to be sore again. If you had a headache, your headache might be worse. If you had, if you don't feel well, you might feel a little worse. So it is probably a good idea, if you can, not to schedule any
big things.
Yeah,
he called
12-hour shifts right after your vaccine.
Yeah, I think it's good. He called in sick, but I just said, hey, I know that little factory is working.
Yeah, but even if you don't get that reactogenicity, the factory is working anyway.
But I was excited. I was like, oh.
I'm next week getting my second dose. I had a little bit of a headache and a sore arm for a couple of days after the first dose, no problem. I want to go back because that question was out of order, Liz. JC, and I like this question for JC. As members of the scientific and medical community, are you pleased with where we are as a society a year later now distributing vaccinations, or do you think that there have been missteps, if any? I mean, we'll all probably chime in on this. I will say, first and foremost, it doesn't matter because we are here now. This is the problem we need to address now. History will judge us. I think history will judge us quite harshly, but it doesn't matter. We have to fix now. Now is the problem.
Yeah. Yeah. I want to say on that note, I think just being responsible going forward with these variants. I do have a slide I would share at the end if anybody wants to hang out. And I talk about like the idea of double masking going forward, because with these variants, we could have another surge this spring. And I am very concerned about it. And it doesn't mean that the treatments aren't going to work right and the vaccines aren't going to continue to decrease our cases. But if people are still out and acting irresponsible about social distancing and masking, we're going to have another surge with the B117. And it's just, the models are already showing where it's going. I didn't include the model graph in my talk because it's not peer reviewed yet, but wow.
Oh,
Yeah.
and you know what? I skipped another one that I wanted to get to as well. And that was from Yo-Yo.
Comment on that though?
Oh, yeah, sorry. Go ahead.
Oh, I was going to say that, a pandemic like this requires an enormous response, intrusive response. I'm from Cuba, and I think we're, as a third world country, the health system in Cuba responded to a different type of infectious diseases in a very intrusive way. Vaccination was massive vaccination. You don't have a choice. for the common good, you don't have a choice. And I think that's what this health system requires right now. A response that is massive, that is intrusive, and a lot of education on prevention. Our health system in this country, it's not on the right direction. We should start with prevention to avoid getting to what we are right now. That's my 2 cents.
I support that 100%. An ounce of, yeah, and we could actually expand this and say an ounce of preparation in 2012 when we saw the second major coronavirus outbreak. If we had funded basic research on vaccines that would so a vaccine that would protect you against all coronaviruses, we wouldn't be in this position as we are now. So I am hoping that we are going to learn our lesson going forward, and we'll actually fund a lot of surveillance work and this idea of making treatments, preemptively making vaccine platforms so that they can be quickly distributed we will have other pandemics. Let me just, as your resident microbiologist, we're going to have more pandemics. I don't know if it'll be during our lifetimes, hopefully not.
I hope.
But, yeah, I think one per lifetime is okay.
It's enough. I've had the experience now. Hey,
Yeah.
I know we have more questions, but there was one question on double masking. I thought I'd throw my slide up on that, if anybody, but the idea of, putting a surgical mask or one of those KN95s and then follow that up with, whatever your cloth mask is. Now, you can have those 2 layers. There's holes, there's little spaces in between. So I think once you do the double mask, that kind of gets rid of some of those spaces, and it provides a little snugger fit. And again, you're decreasing the number of particles that you're being exposed to, and it lowers the infectious dose. So kind of think about that. I think it's a great idea. I've been doing it.
Yeah, no doubt. I want to address a question a while back about thoughts on pushing back a second dose, because I do know, depending where you are geographically, you might not be assured a second dose if you get the first dose, and what are those ramifications. So my thoughts on those, based on what I've seen, really make a concerted effort to get the second dose on time. But if you don't get it on time, if it's, instead of 3 weeks, it's 5 weeks, or instead of 4 weeks, it's 6 weeks or 7 weeks, you're still probably going to be fine. Don't stop, if you hit the end of, I'm supposed to get my vaccine and one's not available, yes, I won't bother. Don't continue to try to find that second dose until you get it, because the boosting effect is still going to be really good. And then on the heels, kind of related, should you get the vaccine if you've had COVID already? Yes. Yes. The immunity you're going to get from the vaccine is going to probably, in all honesty, be better and longer lasting than natural immunity.
I think you could segue. There was a question here. If you had COVID, how long will your immunity last? And it's not.
Yeah.
We don't know.
We know about reinfections. Reinfections are occurring, and you could make the argument of like reinfection maybe occurs if there's a variant change, and we know about reinfections when there was no real variant. It was just you're infected with this, and then later you're infected with this. So without going into too much virology and or immunology, let's just say that oftentimes viruses have kind of built-in mechanisms to subvert the immune response. So if this is the case with SARS-CoV-2, a natural infection of SARS-CoV-2, there could be something within the virus that is suppressing a normal immune response that you then don't get. But if you get vaccinated, you get that targeted immune response. So yes, I think the vaccine immunity is going to be better.
Yeah, that's good. Well, these are really good questions today. I do notice we're at 4:30. I'm willing to hang out for a few more minutes.
I'm willing to hang out. I think Deb might have class.
I know Deborah had class. And I know some of you are getting extra credit for viewing our presentation today. So if you have to go and you have somewhere to go, you're okay in doing that. We understand. We like talking about this a lot.
I like talking about this.
It's kind of fascinating. It's the only it's a terrible thing, but it's interesting to understand it. Deb, are you taking off?
Yes, I have to go. I have a class in 28 minutes until
okay.
Yeah. But it was a pleasure being here with all of you, listening to your questions. And it was a pleasure, Liz and Erin, working together on this project.
Thank you for all of your input.
Thank you.
Have a fun class.
I will.
All right, let's see. Will increasing human Joshua, I feel like your question is the question. So this is like, okay. So normally, right, where do pandemics come from? Why does this happen? And it generally happens when you have something called a spillover event. So you have a virus that's percolating in some animal reservoir, and for some reason it gets into a person, and within that person adapts to being human to human transmissible, and then that one person, gives it to another and gives it to another, and then boom, here we are on lockdown. And so if you have that, and that is where a lot of these things have come around, an outrageous percentage of human infectious disease was once disease of animals that then spilled over into humans. Whenever you have more human to wildlife, where it's like previously not a lot of contact and all of a sudden you have a lot of contact, you're going to have an increased amount of chance that a spillover event might occur. So yes, if the human population is to grow and to grow and grow, related to that is encroachment on wild places where we previously haven't been. And so maybe in this deep cave, in this deep part of wherever where humans have never really been, there's a whole bunch of viruses that are already existing there. If we start encroaching, then you get these spillover events. I will say that with the original SARS outbreak, SARS Classic, that was backtraced to where they could kind of figure out the lineage of that virus. And it turned out that it was likely a coronavirus circulating in a bat population, but in these geographical regions, people go in and harvest the guano, the poop, and sell it for fertilizer. But in this particular case, somehow that virus got into a civet cat, which is a kind of an attractive little wild cat, percolated in the civet cat, and then that was then spilled over into humans. And then, so yes, as human population grows, there will be more chances of pandemic. Because along with that, and Liz, I'm going to give you a second. Oh.
No, I'm fine.
Along with the human population, disease really transmits well when there's lots of population in small areas. So as the population grows, we're going to get tighter and tighter for space. That means our contact with other people is going to be greater and greater. So should something spill over into people, it'll have a better chance of starting a really wild person-to-person transmission.
Yeah, no, the only thing I would add to that, that's all spot on. The worry is when we degrade ecosystems and communities, we take down rainforest areas, we get closer and closer to where we might have animal populations that are reservoirs of these viruses that can then jump to humans. And sometimes in the past, we just hadn't come in contact with this particular virus yet. Now, I don't know, Erin, if you heard this, and I can't remember where I read it or heard it, I should have wrote it down, but it looks like the Wuhan reference virus, when you kind of look at it and look at other coronaviruses, it looks like it probably would have evolved back in 1964. And it's been there, it's been in wild populations. We just haven't come in contact where it had that opportunity to jump over to us. So it's funny because we're like, oh, it's this new virus. Well,
Right. I think with the whole new virus, new to us, right? Yeah. It new, some kind of, when the spillover event of some kind, and I will tell you that there's a team that's currently in China that is looking into the origins of this. And Peter Daszak is part of that of EcoHealth Alliance. And so I look forward for them to finding out, it'll be good to know. But when you have those kinds of things, if anybody's interested in a book specifically on spillover events, it's a book that's beautifully titled Spillover. I can't remember who it's by, but there can't possibly be that many books called Spillover. And it kind of behooves us as humans to take more precautionary steps insofar as to doing a lot of surveillance work, seeing what viruses are out there. If you want information on like how I think we ought to go forward, Google a One Health approach. That involves field scientists going out into caves and wild places and taking samples very carefully, going back into the labs, sequencing what viruses or other pathogens that might be out there, talking with physicians and saying, hey, let us know if you have a certain pocket of infections that are kind of like this, and kind of like everybody working together. And if we kind of can do that and start using these sequences to look for a pan corona vaccine candidate, where one vaccine will protect you against all corona vaccines, looking for treatments that are pan-corona treatments, one antiviral that could potentially, I think that in the future would be a really good plan.
Yeah. Well, and I want to kind of say, I offered extra credit to some botany folks who I know are majoring in biology. And I just kind of want to say, sometimes students are like, well, what is going to else be out there for me to research? Haven't we researched Well,
No.
There's so much. And this just opens up a whole new field of research. We're going to be thinking about this pandemic for years to come. We're going to be picking apart what exactly happened. And then looking at new viruses in the future and the emerging disease.
Virus X is what they call that.
Yeah. So I don't know. There's a lot to do out there. Like, I guess that part's exciting. Like, there's
all exciting. I don't like living through a pandemic, but as a microbiologist with an interest in infectious disease, I'm like, okay, neat. So I see in the chat that a couple of you have found that book. Yeah, Spillover by David Quammen.
Oh, yeah. I have that book. I forgot.
You should definitely. It's a good one. Yeah.
Okay.
And then Alexandra says, what country, if any, do you think we should model as far as helping prevent the spread? I noticed that places like Japan, they have machines that you walk through that sanitize you. So there's no, I'll let Liz, do you want to, I don't think there's not one country other than New Zealand, but you can't model a 330 million population country with the same as a 5 million population country. Like that's like apples and oranges, the things that worked there and they worked beautifully there. I don't know if that would work here, especially because our governments are all different. I will say that the countries that went to mask mandates fast, like Taiwan was like, hey, let's just send you guys new masks all the time. So their residents get governmental purchased masks sent to their houses all the time. And there's a mask mandate. There is no excuse not to wear a mask.
Yeah.
So they never shut down because through mask mandates and physical distancing and saying, hey, if you can work from home, do work from home, to allow the people that can't work from home to more safely go about their business. That would have been great.
Yeah. I'll tell you, there's one story I heard, and I just don't think it would happen here. Americans have our culture is different, but in Australia, you'd think they would be kind of close to us, right? Perth, Australia, about 2 weeks ago, by the way, Perth hadn't had an infection in weeks, like 0. And there was an infection. There was one infection, and they triggered their emergency alert system. And they said, guess what, y'all? Stay home. We were going to figure this out for a couple days. We're going to contact trace this, and then you can go out again. And everybody did it. They nipped it in the bud. So it's kind of funny. There are these little things that, like, if you said that in Modesto, I don't know what kind of response you'd necessarily get. There's this thing here. Somebody put a question. There's places in Japan, they have machines that you walk through and sanitize you.
That's kind of
They might sanitize some of the outside of you. They might have a little UV going. They might have some sort of disinfectant. This just sounds like something that probably is not the greatest. Ultimately, those particles are coming out, those droplets, out of your mouth and out of your nose. They can't sanitize it in there once you've got a population taking over. So, they get your hands, they get whatever. Maybe it reduces the fomite transmission. I don't think it's really I see them spraying in other countries, like with machines going down the street. I'm kind of like, well, I don't know.
So the fomite transmission, so transmission by touching inanimate objects and then touching your face, that is really not an efficient way of transmitting SARS-CoV-2 infection. It really is by breathing in droplets. I don't know if you could make you might want to make the argument, if this is only responsible for 5 to 10% of the infections, fomites, is it worth doing this giant sanitizing? I don't know if the resources. You could also argue that if you saved one life this way, it's good. I don't know about that, but I will say that fomite transmission is really low on the list. And then, so Belen, I see the county told your dad to wait 3 months before receiving the vaccine. So the reason I asked, if you're looking at the chat, the reason I asked about receiving either convalescent plasma treatment for a COVID infection or receiving a monoclonal antibodies is because if you have COVID-19 and you either receive convalescent plasma or you receive one of the monoclonal antibody cocktails, those antibodies are protective. Those antibodies are neutralizing antibodies already. And so you can argue that you don't really need the vaccine for 3 months. These antibodies they don't last forever. They have a half-life of, I think like 1 month or 2 months. And so gradually by the time 3 months is over, they're going to be out of your system. So if you have had antibody treatment, then yeah, the vaccine's probably, you're already protected. If you're just COVID disease, you were just sat at home and felt ill and weren't treated with these different antibody treatments, then I'm not sure why the county said that. I don't know if I subscribe to that.
So one just popped up. This is a great question. After we all get vaccinated and wait the recommended period, is it okay to go camping with a big group of friends? And I would say if your big group of friends has all been vaccinated,
party on,
and the time has passed, go camping.
Party on.
Can I come? That's terrific. No, yeah, absolutely. That's where we're getting, once you get your group and they're vaccinated, like my house here, everybody can't wait. We got one done in here. We're waiting.
You're 1 for 4. We're 1/2 of 2.
Yeah. So anyway.
So, okay, but to caveat that,
yeah.
Joshua, yeah, if you have a group of 10 friends and all 10 of them have had both vaccines, you've waited the 2 weeks after that second shot, and you guys want to go camping together where it's just going to be the 10 of you, party on. Where it gets a gray area, right, is if I'm vaccinated, can I just mask off and go out into the world and party on for me, but y'all have to, stay protected and do your own thing? And the answer is no, because we don't have the data yet. I am hopeful that the data is going to be coming at some point, but we don't have data yet on whether or not or what percentage, I guess, of vaccinated people, if they get infected with SARS-CoV-2 and they're vaccinated, can they transmit to somebody else? And so that data is a bit stickier. But if you know that you're going to be in a group of totally 100% vaccinated individuals and you're just going to go hang out with yourselves, off you go.
Yeah, I think those kinds of activities, and you have your pod, you guys are good. So like if you're heading up to go camping, just wear your masks when you go in stores and stuff in the meantime, right? But that protects everybody else. But you guys as a little group, good to go. So those times are coming.
Yeah. Vaccines. I just want to be vaccinated. Fully. I'm halfway there. Yeah. So another, does the vaccine work like a flu shot and that can help just reduce the severity and not necessarily 100% immune? Yes. And here's the part, biology is all about gray areas. So I imagine that if you had 100 people that took either the Pfizer or Moderna vaccine, whatever, maybe 50 of those people that would be a lot. How about maybe 20 of those people would have sterilizing immunity? And maybe a big percentage of people wouldn't have sterilizing immunity, but protection against any kind of symptomatic disease. And then maybe a couple of people protected against severe disease. So yeah, unfortunately, we don't know who's going to have that sterilizing immunity and who's going to just be protected for severe disease.
And there's another great question just after that.
Oh, these questions are so great. Extra credit for everyone.
Oh, so much. And I love that you guys are still here.
Yeah. Oh, there have been questions. This will be, I assume, Liz, this is going to be available on the Science Colloquium channel, right?
It's going to be able to view. People can view it, share it, watch it again, because I know, it's pretty entertaining.
So would you like to do Julian's?
Oh my goodness. Some people think the government is trying to control us by these vaccines. What is your opinion about that? My opinion about that is that no, the government is not trying to control you with the vaccines. They could if they wanted to with something else. Like they're not controlling you. Nobody's trying to control you. You do have a phone. If you're worried about that vaccine, put your phone in the trash can. Don't let people. No.
Yeah, I'm gonna piggyback on that and just kind of clarify my thoughts on that. You have to think about what's the motivation?
Yeah.
Okay, so controlling how? They're gonna control us. I have heard, the microchip Bill Gates stuff or the 5G stuff, and they want to somehow, and to that, what's the purpose? There is no purpose for that. And scientifically, that's not feasible. And as Liz mentioned, if you have a Facebook account, if you have an Instagram account, if you're on Twitter, Snapchat, if you're on Google, if you have a computer, if you have a phone, those companies already, they know where you go, they know how long you spend there, they can tell, what you're interested in. So it's just why would the government bother?
Yeah.
That's why bother?
The other thing, you guys, is kind of looking at the research out there and vaccine development and the entities involved in vaccine development. They're not interested in that.
They don't care.
And so that's the other thing when you're looking at, nobody's writing in their methods and materials okay, where are we going to put the nano particle? There is no nano. That's not how.
Well, so people get caught up with the word nano, right? Because lipid nanoparticle, and they're like, we got nanobots. Yeah. So it's just a teeny tiny lipid that's surrounding it. That's the nanoparticle part of it. But
there isn't a device in there.
No.
They put it in your In-N-Out burger if that's what they wanted to do. But they're not.
It's just not practical.
It's not a thing. Okay.
All right, any last questions? The 10 countdown?
Yeah, it's pretty great.
All right. Well, Liz, I had a good time.
I had a good time too.
I like talking about this.
Yeah, no, I know. It's fun. I enjoy talking about it, and I know my family does not want to talk about it anymore with me. They just want it to be over.
Yeah, well, same. I want it to be over. I would love to be able to talk about this as, remember that time where we had that pandemic?
I know. It's totally true. Hey, I want to thank everybody for joining us today at Science Colloquium. We're going to be back in 2 weeks. We're going to talk about carbon sequestration on that day. And I look forward to seeing your comments and you guys logging in. If you have not subscribed to the MJC Science Colloquium channel and you want more of what we do in the future, you can subscribe and then you'll get a little alert when we go live with these presentations. We leave all our presentations up on the channel, so if you want to send them off to your friends and neighbors and family, we encourage spreading the science in our community. So thank you so much. Bye.
Spread science far and wide.
Bye, everybody.